Yeah, all right. So photosynthesis. Reminder: the products of cellular respiration will then become the reactants, what we're going to put into for photosynthesis. Meaning, when we did cellular respiration, water was a waste product, and we also had CO2 as a waste product. And so then water, CO2, and then the energy from sunlight is going to be used to then. Main thing here is taking that CO2 and turning that CO2 into a sugar. And then that sugar would then be used, that'd be a product, that sugar would then be a reactant in cellular respiration. Now, they say sugar because, again, it's not technically glucose that you get out of photosynthesis. It's technically something called a sugar called G3P, which can then be converted into glucose. But you can think of glucose, that's fine. I do think it's actually worth pointing out. Notice the formula they have, CH2O. What that's trying to say is sugars like glucose is C6H12O6. Sugars will take the form of 1 carbon for every 2 hydrogens for every 1 oxygen, that ratio. That is actually an important thing to remember, that ratio for sugars. Like, they might give you a sample molecule and ask, like, what biomolecule is this? And you know it's a sugar based on that 1 to 2 to 1 ratio. Okay? All right, anyways, we can break up photosynthesis into what they call the light reactions, and then what you'll see called the dark reactions, also called the Calvin cycle. Okay? Where the idea of the light reactions is we're using the energy from the sunlight to create ATP and NADPH. Okay? The energy stored in your ATP and the NADPH are then going to be used to do the dark reactions, the Calvin cycle, which is where you're going to take CO2 and basically you're going to link 3 CO2s together to create the G3P, the 3-carbon glyceraldehyde 3-phosphate. If you're thinking of like making glucose, you can think of it as like 6 carbons, but technically the Calvin cycle, you're linking 3 CO2s together. Locations, the light reaction is in the thylakoid, so this is the like if the whole stack we call the granum, the thylakoid would be a poker chip. So you can think of the thylakoids within the broader chloroplast as a poker chip, a stack of poker chips, a grand, if you will, of poker chips would be the granum. So, anyways, and then the stroma, that would be like the equivalent of the matrix of the mitochondria. That's where the Calvin cycle is going to be happening. Okay, and then technically a waste product of photosynthesis is oxygen, which is then released into the atmosphere, and then we can breathe that oxygen in, or even plants themselves will need the oxygen to do cellular respiration. Okay, so let's get into some details of this. Get my cursor over here. So light reactions seems a little overwhelming, but if you remember cellular respiration, which we had gone over earlier today, it's really not too bad actually. It's broken down into 2 photosystems. So photosystems are these collection of proteins that will essentially harvest light. They kind of act like solar panels. So there are 2 different photosystems. Light will strike either photosystem. Let's focus on the first photosystem. Ironically, the first photosystem is photosystem 2. They were just discovered in a different order. Now, photosystem 2, the light's going to come in, it's going to hit these chlorophyll molecules. So, not to be confused with borophyll, chlorophyll is these little molecules here. They're going to absorb the sunlight, and then the energy is passed from one molecule to the next. And there is then what they call a primary electron acceptor. So that's what's represented there. That's it's just another chlorophyll molecule. You do want to know the that's called P680. So P680 is the primary electron acceptor in photosystem 2. Photosystem 1, it's a P700. And here's how I always remember it. 2, you can think of 2 as like 20. 700 minus 20 is 680. Or photosystem 1, it kind of looks like a 7. One's kind of look like 7s. it turns out, you know, if you don't think about it too much. Anyways, so yeah, the electrons are going to be passed from one molecule to the next until they then get elevated here, and then they're going to be passed down your electron transport chain here. So they go on down the electron transport chain. So this is fairly similar to what you saw when we went over the electron transport chain in cellular respiration, where the passing of these electrons down these protein molecules will create this hydrogen ion gradient. So the thylakoid space, this is one thylakoid, the interior of the thylakoid space has a high concentration of H+, so that would correspond with a low pH. And those H pluses come from the stroma, that's like the cytoplasm of the chloroplast. So that is why it has a low H plus concentration, because those H pluses are going into the thylakoid space. So this is low H plus concentration, which would correspond to a high pH. Okay, and I'll explain what we do with those H pluses here in a minute. Those electrons will then will meet up along with the excited electrons that come from photosystem 1. So photosystem 1, its main contribution is to get us to do to make NADPH. So photosystem 1 has its own electron transport chain, but its electron transport chain is trying to make NADPH. Photosystem 2, its main job is to create the hydrogen ion gradient. Okay, so make note of that. First, for photosystem 2, it's making, here, I saw it. Check this out. Looks like an H. You know what I'm talking about? 2. I don't know if you ever, I don't know about you, when I draw an H, or I write, I don't know if you draw H's. I do 2, and then so photosystem 2 makes the H's. Anyways, photosystem 1, you make the NADPH, and I don't know what to do about that, you know. Just whatever. It's just the other thing. The NADPH, we're gonna use that. The stored. When you think of NADPH, I want you to think of stored electrons, or stored potential energy, right? So stored electrons here. We're gonna use this to essentially be used to help us attach those CO2s, the carbons from the CO2, to make that bigger sugar. Anyways, going back to those hydrogen ions that we generated from photosystem 2, we have that hydrogen ion gradient, and you got to think of this in 3 dimensions. The stroma wraps around the thylakoid. So here, this is also the stroma down here. Because there's a higher concentration of hydrogen ions in the thylakoid space compared to outside the thylakoid, you know, thinking of diffusion, the hydrogen ions want to move from more to less. Think of a windmill. The energy of the hydrogen ions moving through ATP synthase, the movement of that will catalyze the reaction of ADP becoming ATP. Exact same thing that you saw in cellular respiration. So it's pretty nifty how you have that essentially the same complex there doing both processes. Now water, kind of a lesser detail. We have to regenerate these electrons here because these electrons are getting removed to go through the electron transport chains. So water is the source of those electrons. So, and when you strip water of its electrons, the H pluses, the H's from the water will help contribute to the hydrogen ion gradient here. And then the O from water will turn into your oxygen that gets released from the plant. Okay. I think that's everything. Is there any questions about this? So, anyways, so this is the light reaction. So we're going to use the NADPH and ATP. We're then going to go to the dark reactions, the Calvin cycle. So let's walk through that. Now the Calvin cycle, when I show it to you all like detailed out like this, it seems really complicated. They're not, or they really shouldn't be asking you like, you know, what is made, like sequentially what comes next. What you do want to key in on is the 3 phases. The 3 phases and then like inputs and outputs, kind of like what we saw with cellular respiration up here. So what you're going to put in for cellular respiration, or for photosynthesis, and I'll write it right here. We're going to put in 3 CO2. So that's what's being represented by the gray circles here. So 3 CO2. And you're going to get out, so I'll do it a different color. So an output here is your G3P, which becomes your glucose eventually. To make this glucose, we have to put in 3 CO2. You're going to have to invest 6 ATP. Or I'm sorry, 9 ATP. 6 and 3. So 3, 6, 9. You know that rap song. 3, 6, 9. Okay. For contractual obligations, I can't rap the rest of the song, but you got to put in 6 at ADPH, 9 ATP, and 3 CO2 to make 1 G3P. Why that makes sense is because G3P is a 3-carbon sugar. If I want to make glucose, I would need times 2. I would need 2 G3P. So I have to do that cycle. Well, this whole cycle is actually done 3 times. So like when they say 3 ATP, that's actually representing us doing the cycle 3 times. Anyways, details of the cycle I wouldn't worry about so much. But first part is carbon fixation. That's where you're going to be attaching the carbons to a 5-carbon sugar called Rubisco. I would make note of that. Rubisco is going to be Rubisco is the enzyme. Rubisco is the enzyme. RUBP can be easily confused with Rubisco. RUBP is a 5-carbon sugar. That's going to meet up with 1 CO2. You got to do the cycle 3 times. And that thing gives you your 6-carbon sugar here. And that process there, Rubisco is the enzyme that catalyzes the addition of a 5-carbon sugar to the CO2. That's carbon fixation. And then we are then going to do the reduction phase, where the reduction phase we're going to have to bring in some ATP and then the NADPH. The reduction, the addition, when I say reduction, think hydrogens, adding hydrogens. So adding hydrogens, where the hydrogens came from the H on an ADPH. Okay, so that's why we had to make the NADPH. And then the last phase is regeneration of the CO2 acceptor. So in other words, we have to regenerate the RUBP so we can keep doing the cycle. That's where you're going to have to invest 3 more ATP. Okay, but again, I really think if you can get this 3, 6, 9, and then G3P is what you get out, you're in a pretty good spot there. Questions about that? And if you're wondering, like, do I eventually have to memorize all of that? I never had to in college, but I also went to Texas A&M, so I don't know. Right? Usually, like, if you're doing like medical stuff, they don't really get into the super weeds of this, just because we're not plants, Anyways, this is a really good summary slide here, kind of bringing it all together. So, electrolyte reactions. Remember, there's 2 electron transport chains. The first electron transport chain, what's its main goal? H plus, yeah, because 2 looks like an H plus. Photosystem 2, think P680, because 20 and 2. Photosystem 1, that second electron transport chain, that's where the NADPH is coming from. Okay, that's what we're mainly doing with that second electron transport chain. Technically, inputs for photosystem 1 are NADP plus and ADP. I think it's kind of common sense because you are making ATP and NADPH. And then you're also making oxygen here. Okay. The numbers, there's not really numbers you have to worry about as so much, other than like the P680 and P700, but like the number of ATP, the number of NADPH, you don't need to worry about that because that changes, right? It's going to change depending on the amount of sunlight and stuff. That's not really something to be like calculate, or you would need to worry about calculating for your purposes. Okay. All right, so then the energy in ATP and ADPH goes to the Calvin cycle, and that's where the 369 comes in here. I don't know if they would ask you that. Well, they might actually. The storage form of sugar is starch. That's like we eat starches, like when you're eating pasta. That's because it's a storage form for the plant, is starch, right? If a plant wants to send, so like this is actually helpful to think about. Remember, a plant, only the green parts of a plant are doing photosynthesis, right? It sounds like common sense, but maybe not when you're in the middle of an AP exam. You're doing photosynthesis in the leaves, but the roots also need glucose for those cells to function. So I got to transport that glucose to the roots. But you can't transport glucose throughout the plant. You have to convert it into another type of sugar called sucrose. So I don't know if they would really get into that kind of detail, but sucrose is the transport form of sugar in a plant. Calvin cycle is in the stroma of the chloroplast, so make note of that. Okay. Yeah. So CO2 comes in, O2 comes out. All right. That's all I can think of. Any questions? You good? All right. Well, there you go.